Our next speaker is Andreas Tolias. It's great to have Andreas here. He is one of the newer faculty members of the Stanford Neuroscience community, having joined us in 2024 in April. I think that's about right. He's a professor of neuroscience here in the Department of Ophthalmology. Prior to joining us at Stanford, he was at Baylor College of Medicine and at Rice University. Andreas' lab works on the interface of neuroscience and AI research. He uses AI to construct predictive brain models that link stimuli, neural activity, and behavior with these digital twins. His group conducts in silico experiments that test theories that otherwise are impossible at scale in living humans. Kind of nicely builds on Mackenzie's talk. Illustrating the bidirectional influence of neuroscience and AI, using AI to advance our understanding of the brain, and using neuroscience to build these next generation of perhaps less artificial, more intelligent algorithms. He also is a recipient of numerous awards, including an NIH Eureka Award, Director's Pioneer Award, amongst others. Today, Andreas will be talking about a less artificial intelligence.
Thank you. Thanks for the organizers. It's great to be here, and thanks for organizing this very timely workshop. So are these dogs or muffins? This is something that is innately easy for us, but believe it or not, despite the tremendous progress in AI, this is something that even the more current state-of-the-art algorithms fail to do very well, particularly when you cascade and you create some clutter. And the reason, and it is a well-known problem that is being observed for the last 10 years or so, that artificial intelligence is very good at generalizing or performing within distribution, but once you push it outside this distribution, like in this case, you put dogs and muffins in a composite way that it has never seen before, it fails quite miserably. And the current sort of approach to solving this problem is nicely illustrated by this photo, where basically it's just collect more data. So you make everything within distribution, and then you won't have this problem. And to some extent, it's working fairly well. That's why we have ChatGPT that is quite impressive, and we also, have autonomous driving now. If you go to San Francisco, you can drive around without a driver, and it is a very, competitive, let's say, approach to doing this, but it's also very expensive and very energy consuming, and maybe we'll be running out of data soon. So a holy grail in neuroscience is to really map correspondences between brain activity and structure and our perception, action, and cognition. What in mathematicians like to call isomorphism. You have these two different things like brains and the world and your perception or your actions, and you're trying to find relationships between them. And I will focus on one level for this talk today, which is what we call the representational level, which has been extremely fruitful for the last almost 100 years, starting with the discovery of the action potential by Adria, going all the way to major discoveries, for example, the entorhinal cortex, more recently, that the Mosers did. And what is impressive here is that people recording from individual neurons have found isomorphism or correspondences between things like, somatosensation, all the way to, the representation of space, where a rat is located in a room like this. However, if you look at the history of these discoveries going all the way back to Adrian, and you can read about it on Cambridge University's website, there are mostly serendipitous discoveries. These scientists they had maybe strong intuition about what they were looking for, but as we heard today from Doris, for example, Hubel and Wiesel have this 60-minute review here explaining how they discovered these things. They were using spots of light because they were students of Kuhlfelder who were studying the retina, and one day, accidentally, they discovered orientation selectivity. And if you now look at, let's say, the macaque brain, and we understand the visual system, we have in the order of dozens of visual areas. Within each visual area, we have dozens, maybe hundreds of cell types. It's going to become a very, very difficult process to really try and decipher the neural code with, that classical approach. And the problem lies that the problem is very high dimensional and is non-linear. So for example, if you have a monitor, you can generate 2 to the 65,000 patterns, for example, even on a simple black and white monitor, and this space is too big. And if you take into account behavioral, like we heard from Mackenzie, and you take cognitive states and internal states and so on and so forth, it becomes really, really difficult. So what I will talk to you about today is really what this symposium is about, and we've heard about it, very nice talk this morning, and just now from Mackenzie, is we are in a very special moment in the history of trying to crack the neural code because of two parallel revolutions that really happened mostly in the last 10 years. One is neurotechnology, a lot of it thanks to the BRAIN initiative and other initiatives that have happened in the US and around the world, where we now have technologies, we can record data at very large scales that was not possible 10 years ago. For example, this is a light beam microscope developed by Lipasha Vaziri that can record in the order of 1 million neurons simultaneously. And this is the very well-known NeuroPixel technology developed by IMEC that really has been revolutionary. And on the other hand, we have methods like deep learning that enables us to build data-driven models that can ingest this data and use them to learn predictions and relationships between different types of data. And in the last couple of years, we and others have developed a method, we call it in our method inception loops or building digital twins. And I will describe you what this approach is about and give you two examples of what we think are important discoveries that we don't think we would have discovered them otherwise. So inception loops has four main components. The first is you just do very large scale data recording. This was originally developed in my lab by Edgar Walker, Fabian Sin, and Talia Mohammed when they were in the lab. So the first step is to record large data. In this case, this is an experiment in the mouse visual cortex in layer 2-3 where we record 5,000 neurons, and we show a high entropy natural stimuli. The animal is just running around doing whatever it wants to do. The second step is you use deep learning to build highly predictive models. There's many different ways how to do this. This is just a convolutional deep neural network that is data-driven. Because in a single experiment you have, let's say, 5,000 images and 5,000 neurons, you have 25 million training points, even on a 2-hour experiment. So if you run this for a few days or a few weeks, you get into very large data sets. However, once you have this in silico model, despite that we all think, deep neural networks are a black box, they are not entirely a black box. You can think about it in one simple way. Once you have a very accurate digital twin of a physical system or a biological system, you can do in silico experiments at scale that would have been impossible to do otherwise because of our own lifetime at the end of the day, or the experiment, the animal we're studying, or we just don't have the resources to do it. So for example, in this case here, what you can do is, we can ask the Hubel and Wiesel question and find out what are stimuli that maximally drive these neurons. And when we did this, we found that in the mouse visual cortex, unlike the standard model of what we think of Gabor filters, many of these neurons had different features. For example, they look more exotic. They had these edge type of detectors. Some look like these checkerboard-like patterns, and others had these croissant-like shapes or T-junctions and so on and so forth. And if you search through hundreds of thousands of natural images, you found patches with a certain level of sparsity that corresponded perceptually to these most exciting images for the mouse. But this is just the model could be extrapolating outside this training distribution. After all, these are deep neural networks that I've shown you at the beginning. They can make a lot of mistakes. But the good thing here is that you can run this in closed loop. You can go and take these stimuli now, show them back to the animal, and verify the hypothesis or the stimuli that you generate. That's why we call it inception, because you synthesize the hypothesis or the stimuli from the model. And when you show them back down, you see that these stimuli drive these neurons much, much higher than many other controls that we have run, and you can read about this in our papers. Now, when you run the same experiment in the macaque, you basically replicate something much more closer to the Hubel and Wiesel scheme where you see these Gabor filters. So now what I want to talk, next I will talk to you about two unpublished works that demonstrate the power of this approach. One is studying contextual modulation. As we know, context is very important, not just for perception, but a lot of things we do in life. You can change your behavior depending on context. And for example, here you have these two stimuli, it could be a raisin or an eye, and really it's the context that disambiguates this information. And this has been studied extensively in the visual system, primarily in V1 and in the retina. And the general idea here is that if you have stimuli that are, you show the preferred orientation in the center of the receptive field or the classical receptive field. If you have the same stimulus in the surround, it produces massive inhibition. Okay? And if you have an orthogonal stimulus, it produces less of an inhibition. And there is literally like, dozens, maybe hundreds of studies going back to the 80s doing this kind of work. However, if you now take this type of stimuli, if you say, okay, this is most exciting images for V4, they look much more exotic, they're not easily parameterized. How do you study center-surround interactions? Okay. Now, with this, and this is the work done by Jia-Kun Fu and Katrin Franke, who is here, what they did is they built a model that was very good at predicting center-surround interactions or contextual modulations to natural images. So they just threw in a lot of images, natural images that were masked with different masks, and these models became very good at predicting the modulation index to unseen combinations of images. Okay. But once you do that, now you can find the MEI, and in the model now, you can do a second optimization step. The way these things work, you optimize it under some contrast constraint of the RMS, the amount of power basically you have in the contrast of the image. So once you find the most exciting image, you can cut the gradient in the center and allow further optimization in the surround by adding more contrast. So it's a severe conditioning the response and saying, given that these neurons are going to be seeing their most exciting stimuli in the classical receptive field, what is a stimulus in the surround that will excite them further or inhibit them? And when Katrin and Jia-Kun did this, they observed something that was very surprising, that the models were predicting that stimuli that seem congruent with the center, which is opposite to what the story that I told you earlier, would excite them further, and stimuli that broke the congruency with respect to this sort of qualitative perceptual thing here, it would suppress them. Okay? So of course, again, this could be an artifact of the model. So they went back and tested this. They created the stimuli and showed that indeed, when you add these synthesized images that look perceptually to us more congruent, they excited the neurons. And when you added these inhibitory ones, they inhibited them. And then they went further and quantified this sort of perceptual congruency by looking at natural scene statistics. If you look at, for example, a patch like that, this is a natural image, and this is the most exciting image. You find that there are most exciting natural images that look like the most exciting image. And if you look at the surround of that, you see that there is this sort of concept of congruency or collinearity in this case. So what we can do here is that we can take the most exciting images, compare them to natural images versus the inhibitory, and we find that the most exciting ones, the ones that have surrounds that excite the neurons further, are more congruent. Now, this created a discrepancy because it could have been that most of the studies that I've shown you earlier were done in macaques or cats originally. And we've done this in mice, but then we used a very different method on how to study the center-surround interactions. The previous studies used parametric stimuli. We use these image synthesis methods. So to figure out this discrepancy, what we did is that we replicated these experiments both in mice and monkeys, and we showed the grading stimuli. It's on bioRxiv, the paper, you can read about it. But basically, we found that even in monkeys, when you use, you look at something that looks like a Gabor, but you see sort of may have some curvature here. But if you do the same image synthesis way, you start getting this collinearity. So you get a very different picture of what exciting stimuli are. And in fact, this is more related to some earlier work done by Charlie Gilbert and Thorsten Wiesel, where they describe sort of more collinear patterns like this. So the second study that I want to show you, this is not published anywhere, not even on bioRxiv, it's very new, and I would like to get your feedback. It's a more recent work that is led by Katrin, Nikos, Konsti, and Sofia, where we're studying area V4. And our area V4 is sort of a middle-level area, lower than the areas that Doris talked this morning, but it's sort of in the same part of this ventral stream. And what we find here is that if you take these neurons and you build a digital twin and you run hundreds of thousands of images through them, or you use the image synthesis methods, you find that their most exciting images are very interpretable, which may not be that surprising because other studies have shown that they're selective for color or textures or shapes. For example, here you see a neuron that likes these like zebrafish-like textures, but it's invariant. These have they don't just have a peak, they have a ridge or a manifold, and they're invariant through transformations of these textures. Sometimes like rotations, scales, and even different shapes, okay? And you see another neuron that likes red, but it doesn't really, and it sort of likes red in a segmented way, like segmenting an object higher up than lower up, but it sort of, that doesn't have much texture selectivity. And here's another neuron that has very strong, selectivity to texture, but invariant to rotations or different other kinds of invariances. So when we look at this, because we have a digital twin, we can show millions of these images, right, in silico. You just need GPUs to do this. There's no experimental limitations. And what we found is that many of these neurons are not very sparse. If you look at well-isolated neurons with depth recordings, many of them are not sparse, okay? And when you show, for example, in this case, 250,000 images, you find that there are some that are most exciting, as I've shown you before, but there is like a group of them that you can search through when you show these, hundreds of thousands that are most inhibiting. And if you look at those, they also seem very interpretable, but almost like a little bit unrelated to their most exciting. For example, this neuron here that like these red patterns, it had this more like, almost like this one that was the most exciting. For this type of pattern, was most inhibiting. Okay, so these are neurons that have spontaneous activity, not very much, let's say in 100 millisecond bin, you have like a few spikes, maybe 5, 10 spikes. But then if you search through main images, you find a few that will drive it down to 0. So the classical view that we think of tuning is that we have this feature space. If this is like the perceptual space or the latent space, we have a bump. This is a neuron. This neuron here. If there's a combination of feature 1 and feature 2, and maybe you can think it's some invariant in some other feature, it gets maximally activated. And then we tend to think that there is, this would be like a more feature detector. But what we find, at least the majority of these neurons that we have good models for, they seem like that. They have one area that excites them a lot, and they have another part of feature space that inhibits them. And this could be, and if you look at this is, for example, another neuron, okay? This is like a schematic of this concept here. Now, this bipolar coding, right now we just call it bipolar coding, could maybe enhance information coding. And this is something that we're working with Sofia Sandborg to, derive some theoretical work, but we'll be very interested to talk to a lot of people here in the room about it. So another thing that is kind of interesting is that if you look at this, if you look at many neurons, let's say hundreds of neurons or thousands of neurons in different models, you see that these directions or these most exciting and inhibitory bumps in this feature space are not just they equally tile the space. They seem to form functional clusters. And this is shown here by the analysis where you take the most exciting images and the least exciting images, you show them to other neurons, and you find these corners, which means that essentially, let's say in this room, some of you like red stimuli and others like green. There's not many of you that like yellow. So there seems to be like there's a shared axis of tuning in this sort of perceptual manifold that we're still trying to understand. And this seems to be ubiquitous. It's not just something special in V4. If you do the same in V1, again, you find the same thing in V1. You find that there are a lot of neurons in V1 have spontaneous activity, and there are, if you search through a lot of a very large stimulus set, you find a set that basically shuts them down, okay? And these seem to be interpretable, and again, you get the same sort of grouping that we saw earlier. Now, Katrin and Konsti and Nikos have a poster, I think it's right here, and you can go and learn more about it if you're interested. So to summarize so far, I think this era that we're living where, and this is not just true, I mean, basically if you take, let's say, AlphaFold or all the stuff that is happening in molecular biology and in biotech, they're just utilizing any sort of approach that you can collect large data set. you can just ingest them with these very powerful neural network models that then enable you to do in silico investigations to optimize or to make discoveries that then, if you set up your pipeline or your platform the right way, you can test them back and prove them or disprove them. So I think it's a very powerful thing. So just for the next 10 minutes, I want to give you like a road ahead of the way we are trying or we see this approach going. And the first one is this idea of foundation models of the brain, right? You can think that basically you have these different modalities, whether it's transcriptomic information, behavior, stimuli, activity in the brain. And on the other hand, you have tasks that you can think of them as like, controlling activity, diagnosing a disease, maybe predicting treatment efficacy, testing theories, or making discoveries. And once you start collecting this data, even if you don't have them simultaneously recorded across all modalities, there are ways that you can stitch them together and create these very powerful models that now enable you to do new types of experiments. For example, here is some recent paper that was done by Eric Wang in my lab. That he started building these foundation models that have dynamics. Here the animals are watching natural movies. They're moving on a ball. He's combining experiments from many, many animals to build models. And I'll show you some of the powerful new things that these models can do. And this is another related paper by Blake Richards, Eva Dyer, and Guilherme's lab in Montreal. So this model is very similar to the one before. It's a deep convolutional neural network, but it has some recurrency and is trained on many, many mice. Okay? And Constantino is building one that's going to get trained on 1,000 mice, I think like 3 million neurons. So these models can scale up a lot. And now, one of the things that you can test in these models, once you have these models, you can feed new mice or new data with very few data. Okay? So you can take a new mouse and you don't have a lot of time to do an experiment. For example, here, you can take a new mouse, once you have this foundation model, and then with 16 minutes of data, you can reach very good performance that would have taken more than 76 minutes to get about the equivalent level of performance. So they show this transfer across mice and across new neurons. The other thing that is quite remarkable is that these models, once they get trained on a lot of data, they start showing capabilities to predict responses outside their training distribution. For example, these models was only trained on movies, and then what Eric did, he created like other stimuli like static images, got bored things moving around, flashing dots, ping noise, these coherent random dots that had optic flow and so on. And this model was not just at predicting the responses to natural movies that it was trained, but also other kinds of stimuli. So now it opens up the repertoire of experiments that you can do. You can also use this is pixel level reconstruction. This is showing the data, showing a new movie to the animal, taking the activity and then passing it, inverting the model basically to reconstruct the pixel level reconstruction. Now these don't look great because it's low resolution, because the mouse visual system is basically lower resolution visual system, but you can see that they look very good in terms of, if you basically, I don't have it here, but if you filter these movies, they look very similar to what we're reconstructing. The other thing that this model can do, which is quite, interesting, is that there is a very special data set that we collected a few years ago as part of this IARBA project, this Microns project. And this is a very special mouse where we imaged about 100,000 neurons. We sent it to the Allen Institute. They did electron microscopy, and then Sebastian Sang reconstructed part of the connectome. So in the same mouse, we have about 60,000 neurons who have alignment between the functional data and the anatomical data. And now based on this analysis, the Allen Institute, on the anatomical side, they characterize the pyramidal neurons from layer 2 to layer 6 as different cell types based on morphological and nuclear characteristics. Okay? And now what Eric did is that he took those morphological classes, and based on the functional model itself, the model never was trained on anatomical data, it could predict, because, for example, there is layer differences in the functional properties of these neurons, but even within a layer, he predicted fairly well the anatomical properties of these neurons. So this is starting to look like you can start combining this data where these functional models can start predicting things that are beyond just function potentially, which makes it, moving towards more of these multimodal models. So the other thing that once we have these models, what we really want to do is understand how the brain, what is the correspondence between the brain, as I said, and the world, okay? So we want to and what we're looking for really in science, and this is a little bit AI or neural networks, is we're looking for symmetry, simplicity, compression of explanations. I mean, that's what sort of we scientists are looking for, right? Trying to find simple rules that can explain these complex phenomena both in the brain and the world. And what I'm advocating for here is that going through this digital twin approach can be very powerful. And I'll show you one very recent thing that we just started doing, again with Liam Nigos, Sofia, Nina Miolaine from USB, and then Catherine Franke. And this is the idea of equivariance. So it's a very it's a very simple concept in a way, but if you take, for example, the shoe and you rotate it around in a circle, you have a rotation, let's say 2D rotation in the real space, like in the world space, nothing to do with the brain, you just rotate the shoe. And then, the question of equivariance is there an equivalent transformation that is happening in neural space, okay? So the idea is that if something rotates in the world, do you see a rotation in neural space? And this is sort of the concept of equivariance. And this is what we think the brain is trying to do. So for instance, here is a case that a little bit surprising. If you look at it in world space, even in pixel space, if you take a shoe and you just rotate it around and you don't take the texture, this manifold is still equivariant even in pixel space without having the brain. So these kind of problems, they seem to be actually trivial, some of them, even in pixel space. There isn't anything interesting going on. But now if you take, for example, these textures, these are made out of, these are 70 textures that are different classes of textures. And now you create these metamers by an algorithm developed by Portilla and Simoncelli. And now you generate these metamers and now you rotate them around. So these are just textures of a certain class. And this is another class, this is another class. I'm just showing you this class here. Then what you see is that in the data manifold, in the pixel space, you lose this equivariant relationship. And if you look at an untrained network, a digital twin of before you train it, so it's a deep neural network with randomly initialized weights, then you don't see it. But once the digital twin gets trained, now you start seeing this equivariant structure. And what you see here is this entanglement of the texture class and the rotation, which is what you expect, and that's what you see, and that's sort of what would be an equivariant relationship between the world transformation here, either changing the class texture or rotating the texture. And what Liam did here, he fitted a rotation matrix to this, and then he showed that, if you take an untrained network, it has it's a chance basically, but if you now take a trained network and you run the rotation matrix, then you can, give any rotation and you can predict, how it would rotate. You can rotate the texture in the real world, and you can predict how it would rotate in the neural space. Now, this type of thing may not be that surprising because these textures have very strong orientation biases and orientation symmetries, and we're trying to even do it in V1. I think even maybe complexes in V1 are possibly capable of doing this type of transformation, but I'm just trying to show you the approach that we're taking. And then what Nigos did here, he's basically, if you learn, let's say, how to decode the orientation, because it's a circle, you can do linear decoding by taking the sine and the cosine, then you can show that if you learn it on one texture, you can generalize to any other texture that has the same type of symmetries, in this case, this one, 1D symmetry. So to wrap up, so generalization, the hallmark of intelligence is really generalization and the ability to generalize outside your training distribution. Like a child, the first time it sees a pink marble elephant, she doesn't just freak out because it knows how to disentangle these latent variables. And this is really like the big gap in current AI. They're not very good at generalizing, although you may think they are because they're trained on such massive amounts of data. So it's kind of, and this is an open debate in the field, how well these scaling laws are going to keep improving. What I think a complementary approach is to try and understand the brain at this representational level, and a lot of these methods can be applied in humans too, all the way to humans. And then if we do like a more global mapping of the brain, we try and align this or try and understand the encoding of these neural manifolds and how we can align it to the brains, that could be very, very powerful. Now I would like to finish with this very nice thing by Daniel Dennett, who came up with this sort of concept of competence versus comprehension. And the idea here is that these structures were built by termites, and this was designed by Antoni Gaudí, and they look very similar. So they, both the termites and Gaudí, they developed structures that to some extent look kind of similar, but there is this concept that there is something fundamentally different, and maybe, though I'm not 100% sure, it could be that current AI, we can think of it more like having competence, but being more like termites, where our brain and animals' brain is more like Gaudí, where it has some comprehension and understanding of the world. Thank you.
All right, that was great. We have time for questions. I'll start off with one maybe then. So as you've worked with these models and both use the models to sort of maybe generate optimized stimuli or figure out the optimized stimuli to sort of drive neurons, and then gone back and forth between the models and the data. Going back to Doris's talk earlier, a lot of the models you're using, I don't know if they have any recurrence in them or any top-down feedback. Can you comment on your experiences as you've been working with these models and going down this path as to whether there are breaking points because of the lack of recurrence or top-down signals?
Yeah, that's a great question. Most of these models that I described so far, they are static. They just predict average activity, mostly to static images. The more later model that I showed you has some recurrency because it's predicting dynamics. And right now, and we tried different architectures, and the concept here is that if you use architectures that are more brain-inspired, they have a better inductive bias, they should be able to perform better. However, practically speaking, there is a more like technical issue that if you use deep learning, you tend to use architectures that they've been optimized, or there's a match with the learning rule, like backpropagation, the way you implement it, how to train them. So it may be that, let's say, if you create a more biologically faithful model, in theory it could perform better, but in practice often it may actually perform worse because you don't have the right learning in the way to optimize it, basically. But I think this is like ongoing work that eventually, we're very interested to explore.
Andreas, so cool. So I guess I'm a little surprised by the bipolar mode of mutual suppression. I guess it doesn't have to be another set. It could be another 5 sets of things that comes, that suppresses this set.
Yeah.
So is the bipolar mutually suppressing? So if one is suppressing the other MEI, does the reverse also apply? Does that MEI also suppresses the least?
Yeah, so what seems to be happening, and this is preliminary ongoing work, is that if you have, let's say, latent space, you have variables, let's say, color, shape, texture, depth, you get these like bumps of, if you plot all the MEIs and all the LEIs, they occupy certain parts of space. But let's say what is exciting for you, it may be suppressive for me and vice versa. And right now there isn't like an obvious relationship between the excitatory and the inhibitory. Now, there is work done by Cornelia Schwartz and Simoncelli and others that show that, some of these models, there's always divisive normalization or inhibition. What's the role of inhibition? And many of these models seem to have selective inhibition. So it may not be that surprising that you have if you have a neuron that has some spontaneous activity, there is selective inhibition. But they seem to be organized that, let's say, what may be the most exciting for you is the most inhibiting for me, and then vice versa.
Hi, Andreas. Brian Wendell from Stanford. Hey, so glad you're here. One of the things about the way we think about the neural coding and vision has to do with the topology, the local connections, and what you're near. And also, for example, take the case of color. It's not just the local connections, but you know it's an L cone and an M cone, and it's not all kinds of neurons are the same, but the L cones and M cones have to be compared with one another, or if it's a spatial pattern in V1, you need to know, it's a neuron to the right of me, it's a neuron to the left of me, and you need to know something about the topology. So I'm wondering, as you're thinking about your digital twins, is there a notion in which there's a kind of a labeling of certain types, or there's or that there's some spatial topology, so that I know that this neuron, actually, when I train it, it's got stuff that's near it, and it uses that information?
Yeah, that's a great question. So, of course, this goes back to this concept of cortical columns, right, going back to Mountcastle. I haven't shown it, but we have actually a paper on bioRxiv by Consti, where if you take, let's say, V4, and you say, okay, is there an organization, a topological organization? Often it looks random, but if you look at these MEIs, they seem to be of the same class, but under some different transformation, for example. So there is this, let's say this concept of cortical columns, and what Doris found with the face cells may be more ubiquitous across the entire visual system at least, but we just have to optimize the right stimuli and map out the tuning function, and this is the advantage that this digital twin enables you to do. So for example, you can just record from orthogonal to the cortex in V4, look at the presence of these neurons, and usually it's 100 stimuli or your favorite set, you just don't find a structure. But when you do it with these optimized methods, then a structure emerges. This paper is on bioRxiv, I can send it to you.
Fascinating talk. I'm particularly fascinated by the termite mound and Gaudy. How long did it take the termites versus the hundreds of years for Gaudy?
Yeah, so I have a good, it took, the termites built it faster, but there's something even more fascinating that I learned from Terry Zinovsky, and he writes it in his book. So during the Industrial Revolution, for many years, they discovered they could build steam engines, but they were not safe, and they used to blow up all the time because they didn't have a scientific understanding of thermodynamics or what's the relation between pressure and temperature. The same is true for the Notre Dame. The Notre Dame, it took them about 200 years to build, and it took the Parthenon 19 years. And a lot of people think that it's exactly what you said. They lost the sort of scientific understanding. For example, the flying buttresses were built sort of by trial and error. So a lot of, and there is this analogy that maybe, AI, it's sort of at that stage now, that once we have a deeper scientific understanding and more like the models that Mackenzie was talking about that are more theory-driven, maybe they will be you get safer AI and better AI. But yeah, the termites built it faster than Gaudy. I don't think it's finished yet.
We have time for a final question if there is one.
Fascinating. I'm impressed by the last page. Obviously, you definitely ponder about human, machine, animal. What's the boundary?
Yeah, I mean, I think that in perception, for example, a lot of primates, the perceptual system is very similar to ours. So there is, but there's definitely obviously language and other things that are beyond animals, right? I mean, that's clear. And more, I think, at least the way that I think of it is I'm not an expert in this area, but I think of it like our brain has a model of the world, and humans basically, maybe using language, have extended this model in space and time. We can think, I don't know, at the beginning, what happened at the beginning of the universe, whereas, lower animals, they have a more restricted world model in terms of space and time and feature complexity. But I think there's more experts to answer that question here.
Okay, great. Let's thank
Thanks.
Andreas again, and Thanks again also to Mackenzie.